Olá, pessoal. Boa tarde. Muito obrigado pela participação de todos vocês nesse seminário à tardinha de sexta-feira. Hoje, essa é a primeira live que a gente vai realizar do nosso grupo de pesquisa do Report Talk. Ela está sendo realizada em conjunto com a disciplina PBA 5016, Seminários em Biociência. Tá? Hoje eu tenho o prazer de chamar para palestrar para a gente o professor Jerry Bouma, que foi meu orientador de doutorado. Então, para abrir essa série de seminários, a gente vai iniciar com o professor Jerry Bouma. Eu vou, então, agora mudar de português para inglês e a gente vai iniciar a nossa conversa. Tá? Eu vou pedir que, ao longo da palestra, vocês enviem as suas perguntas pelo canal de perguntas ali, pelo bate-papo do YouTube, e eu vou colocando elas aqui para responder mais tarde. Então, eu vou trocar para inglês agora e a gente vai iniciar a nossa conversa. So, thank you, Dr. Bouma. I was just introducing how the live will work. And so, first of all, thank you for accepting the invitation. Yeah. It's a pleasure to have you here as our first speaker for this seminar series that we hold. We hold a couple lives on Instagram, yeah. But on our YouTube channel, this is the first talk.
Oh, I really appreciate it. Thank you, Gilles. I really appreciate this. This is quite an honor.
Thank you. So I'll just introduce you first, and then I'll give the word to you. I told the students that they can ask the questions through the chat, and then I will place the questions in the screen so we can talk. Okay. Also, Dr. Bouma mentioned that if you guys want to ask questions in the middle of the talk, it will not be a problem to stop the talk a little bit and answer the questions.
Yeah.
Okay. So Dr. Bouma, he received his PhD in reproductive physiology at the University of Idaho in 2003. And then after that, he moved to a postdoctoral fellowship in molecular genetics at the Jackson Laboratory between 2003 and 2006. Then after that, he got a faculty position at Colorado State University in the Department of Biomedical Science at the Animal Reproduction and Biotechnology Laboratory. His lab currently focuses on the role of epigenetic regulation of sex steroid receptor signaling in placental development and function, which has a significant impact and application to livestock and human health. Besides impressive researcher and a professor, Dr. Bouma has really good grades from the students, from undergrads and graduate students, but also as a supervisor and a mentor, I can say, because I was his first PhD student.
Yes,
I can say that he's one of he was the greatest mentor I had in my entire career, and I had really good mentors in my master's and my undergrad, but he made the difference. Thank you. I'll just give the word to you, and you can start the talk. Thank you, Dr. Bouma.
Thank you, Juliano. Thank you for the really kind introduction. Yes. It's very exciting that, the recognizing now that Juliano was my first PhD student at the same time, that also makes me feel really, really old at this point. But again, yes, Juliano came here a long time ago and was able to start as a research technician, I believe, first, and then subsequently as started this PhD degree, and it's been a tremendous ride, and we've really enjoyed lots of great moments together. So I really appreciate this opportunity.
just before you start, Dr. Bouma, I forgot to ask you just to mention a little bit about your lab and also about the university before you start the talk. Just sorry for interrupting.
Yeah, no, that's good. Absolutely. No, thank you. No, yeah. So I love that. So I started here at Colorado State University in 2006, and I was hired here as an assistant professor in the Department of Biomedical Sciences, and especially in the group of the Animal Reproduction and Biotechnology Laboratory. And so that was, 14 years ago. And since then, as any professor here in our department, we have a kind of an interesting effort distribution in terms of what our job entails. So for me, it was primarily research, but also teaching was going to be a significant component, and service and outreach. And I truly can say that all these three parts have been a tremendous rewarding part of this job. I truly enjoy teaching undergraduates and graduate students. But I really love about this job is the ability to do research and to continuously train students. Students like Giuliano and many others afterwards, and ones that I have right now. And so what I thought I would do is, so today I picked a title that is a little bit descriptive, maybe. I was thinking about talking a little bit more about how the projects, or kind of the different projects that kind of got us going into studying the role of androgens in pregnancy. So today I focus on sheep as an animal model to study the role of androgens in pregnancy, but that's really not how my lab started here back in 2006. So when I came here from the Jackson lab after a 3-year postdoc, really my research interest was developmental genetics, mouse developmental genetics. Specifically, I was interested in genes that were important for fetal gonadal differentiation. So what transcription factors and genes are important for ovarian development and testicular development. And so when I got here, really I got to finish up a lot of these studies here at Colorado State University, and published some nice papers on the role of transcription factors in gonadal development. Giuliano's helped me out with some other mouse models to look at the role of GATA4 in ovarian development as well. But the nice thing of working here at Colorado State University is the opportunity to work with other animal models. And actually, Giuliano and I got to work on things that I'm sure for those of you that have followed Giuliano's career since he left here a bit, we've got to work a little bit on cell-secreted vesicles. And actually, this was all Giuliano's idea. At the time, we were interested in looking at small RNA molecules as potentially a marker for oocyte quality, using the horse as a model. So we had old aged mares that had poor oocyte quality, and younger mares that generally have good oocyte quality. And Giuliano was interested in a project to looking at microRNAs that could predict which ones are associated with good oocyte quality. And I don't know how we got to talking about this, but he was interested in finding out if maybe these microRNAs in follicular fluid were packaged in cell-secreted vesicles and things like exosomes. At the time, I didn't really believe that this was the case, but I gave Giuliano all the freedom that he wanted to explore that, and it developed into this really nice paper in Biology of Reproduction. And I think Giuliano's done quite well for himself, taking that and further exploring the role of extracellular vesicles in reproduction and transferring material from between different cell types. So that was quite a switch for me. I'd never worked with horses before. I'd never done anything with exosomes before, or microRNAs. But I was interested in looking at fetal gonadal development in other animal species. So we explored together with my first master student, Katie, and Giuliano was on this paper as well, to looking at microRNAs potentially in their role in fetal gonadal development in sheep. We use sheep as a model a lot here because they're cost effective. The relevance to agriculture as a model for example, the cattle industry. So we decided to look at the sheep fetal gonads and see what kind of microRNAs were present. So how do I go from that to androgens? Well, that really started with a small pilot grant that we got funded here back in 2008, so 12 years ago. So this was submitted in the spring of 2008, and we were interested in fetal gonadal programming. So at that time, the idea was that if we could find an animal model where that involves some developmental programming abnormalities, to look and see if small RNA molecules like microRNAs, and see if they were, one, expressed, and two, if they were somehow involved in regulating fetal gonadal gene expression. So the goal of that proposal was really to test the hypothesis that prenatal treatment of testosterone affects fetal gonadal programming by changing expression of microRNAs and things like that. The reason why we chose prenatal androgenization or prenatal treatment of testosterone was because somebody in Michigan, Dr. Vasantha Padmanabhan, had already worked with this model for many, many years. And basically what this model is, it's a model to develop sheep that have polycystic ovarian syndrome. So what you do in this model is you breed sheep, and once they're pregnant at gestational day 30 through gestational day 90, and again, gestational length here would be 150 days. But during this treatment window, you give them daily injection or biweekly injections of testosterone propionate. This is a very kind of an artificial model because the levels of testosterone in these animals become really, really high. However, what you end up doing is these male and female fetuses are now exposed to a very adverse in utero environment, and the female offspring that follow from this abnormal programming, when they grow up, they all have polycystic ovarian syndrome. And it's really a well-established model. It's funded by the National Institute of Health for many, many years here in the United States, because it really, those animals, those female offspring, really resemble all the symptoms of PCOS that you also see in humans. And basically what PCOS, really what it affects, or what the adult PCOS phenotype is, it really relates to the metabolic syndrome and a reproductive syndrome. Okay, so what happens here, this could be either to genotype or environment, but basically we did this artificially. We treat these pregnant sheep with lots of testosterone during that window of development, during pregnancy. And this adversely affects the pancreas and the hypothalamus and pituitary. And not only the pancreas, but it also adversely affects muscle, liver, the adrenal gland, and the ovaries and the testes. Okay. And basically when these females then grow up after birth, exposed in utero to lots of testosterone, they tend to develop type 2 diabetes, so hyperglycemia, and are anovulatory. And in fact, the male offspring tend to be subfertile. A lot of these issues are due to abnormal insulin response by beta cells, leading to hyperglycemia, insulin resistance, and on the reproductive side, there is reduced negative feedback on LH. So basically, the ovary starts secreting more and more testosterone because of elevated LH, really reflecting what we see with women that have PCOS as well. Okay. So when we did this model, this sounded really good because it's a very straightforward model. All you have to do twice a week is give them a high amount of testosterone. The reason why these fetuses develop this PCOS is because this treatment window actually covers a period during development when the hypothalamus starts developing, when gonadal differentiation occurs, and when the pancreas starts developing. So this developmental period from gestational day 30 to 75 or to the 90 really impacts the hypothalamus, the ovaries, and the pancreas. So we thought this model was developed, is established. So let's give this a try and see if we can look at an abnormal developmentally programmed ovary. So we started this and asking the question if we can induce developmental programming of ovarian dysfunction. So our first experience was that we started dissecting gestational day 90 fetuses. On top here, you see a control fetus with the presence of teats in these day 90 fetuses, female fetuses. And what happened was in females that are treated prenatally with testosterone, you'll see development of a scrotal sac. So clearly these animals, they will not have testes. They'll still have ovaries. They'll also have the teats still present. However, they'll develop this scrotal sac, which is really indicative of prenatal androgenization. So we collected the fetal gonads and we did a microRNA screen looking at lots of microRNAs. And for a variety of microRNAs that we looked at, we wanted to see if they actually impacted genes we know are important for ovarian development. So in blue here, we have estrogen receptor alpha, beta. We also looked at DNA methyltransferases and their expression in fetal ovaries. And in all cases, we saw a significant increase in their expression. We also did a little bit, because of the DNA methyltransferases, we were wondering if the estrogen receptor maybe is methylated. So we did a chromatin immunoprecipitation assay and looked at the estrogen receptor alpha promoter in the ovaries of gestational day 90 fetuses. And we were able to pull down methylated estrogen receptor alpha promoter using MECP2, which is basically a methyl binding protein. So this was all really interesting. We wanted to look at microRNAs because, again, one of the things that we learned during, or at least that I learned during my postdoctoral fellowship at the Jackson Lab, we did a lot of microarrays and gene expression analysis, trying to identify all the genes important for ovarian and testicular development. And I guess it's kind of good, but frustrating at the same time, because there are hundreds of genes that are upregulated in fetal testicular and fetal ovarian development. So one of the things that we started talking about, in order to regulate this correctly, we think that microRNAs probably play a role in this. This is just from a review paper. I think most of you are familiar with microRNAs. Again, if you followed Giuliano's work over the years, he's quite the expert himself on microRNAs. And these are these small RNA molecules that will bind 3' UTRs of transcripts. And in fact, what you then can end up happening, several microRNAs can all have the same targets. You can have one RNA target that is regulated by 5, 6, 10, 20 microRNAs in such a way that now we affect many, many different biological processes. So we thought, okay, if you have so many hundreds of genes differentially expressed between fetal testes and fetal ovaries, microRNAs are probably a great way to do this biologically. So we did a lot of screening. We did a lot of microRNA profiling back then, as a lot of that is published in one of our earlier papers. And this is just an example of 2 microRNAs where we saw a decrease in microRNA 200B following prenatal testosterone treatment. And this microRNA 200B is predicted to target known genes that are involved in testicular and ovarian development, such as GATA4 and FOG2, VEGF, SMAD2, and similarly, microRNAs that were upregulated that affect different genes that are involved with ovarian or testicular development. And at that point, we kind of I kind of hit a wall with the microRNA stuff, because we've identified these microRNAs and their potential and predicted they could play a role in fetal testis development or fetal ovarian development. However, at that time, we weren't sure how to study the function of these microRNAs. So there were really 2 problems. One, you had one microRNA that could target so many genes. You also had many microRNAs that impact the same gene. And then on top of that is, how were we supposed to knock out some of these microRNAs? That technology back then wasn't really there. This was really almost 10 years ago, when we didn't really have many microRNA mimics or overexpressors or anything like that. Plus, remember that all this work was done in sheep. To make a knockout sheep, that still is barely feasible. So at that point, I decided to leave this area alone a little bit, primarily because I really didn't know how to further pursue it. Looking back, and as I was putting some slides together, the field of microRNAs in reproduction has clearly advanced. This is a review paper from a year ago, highlighting all the different processes in reproduction and the microRNAs that have been identified. Now, the very critical amongst us will recognize that a lot of these microRNAs are probably just indicated here based on their presence. There are very, very few studies that in fact actually deleted microRNAs to show a function in any particular process in reproduction. So the field has definitely come along of quite a bit. We know that microRNAs are very important in reproduction and development, but I think there's still a lot of work to get done. So how do I get from this to androgens in the placenta? Well, let me take you back to that prenatal androgenization model, this PCOS model in sheep. As you can remember, when we started collecting the tissue at gestational day 90, the first thing that we would see is the placenta. Okay, so here's a sheep uterus on the left side containing a fetus. And for those of you that are familiar with what a placenta looks like in ruminants, these buttons here, the placentomes, that is the placenta. Okay, so in a control ruminant, in the case of sheep, you'll have anywhere between 75 and 100 of these placentomes. And they're basically that's the unit where maternal and fetal interface occurs. So this is where the exchange between mom and the fetus occurs. Nutrient supply, oxygen supply, getting rid of waste. So instead of in humans where we have one disc, in the sheep and cattle, we have all these distinct placentomes. I have to be honest, I had never, up until that point, seen a sheep placenta before. I had no idea what to expect, and I had no background in placental physiology whatsoever. But that's, again, an advantage of working in academia and in science. If you see something interesting and you have an opportunity to study, you go for it. So when we looked at that prenatal androgenization model, we had our control sheep. We saw all these beautiful placentomes. And in fact, what that placentome looks like here on the right side is basically the maternal caruncle, which is kind of the outside, and a really dark inside, which is the cotyledon. Okay, so that's the fetal portion. So mom's portion is on the outside, and on the inside we have this the fetal portion. That's usually classified as a type A placentome. What happened when we did that prenatal androgenization in sheep, we started seeing placentomes that look quite different. So we not only had type A placentomes, we had something called type B placentomes, type C placentomes, and type D placentomes. And what basically happens, this is a nice cartoon picture on the right, that you see a shift. So the white here, this is looking from the side. The white part is the caruncle or the maternal tissue. The dark side is the fetal tissue or the cotyledon. And if you go from A to B to C to D, you could see that it looks like the cotyledon tissue starts to overgrow and take over. Okay, so what we noticed what testosterone treatment did for those 60 days is a shift from type A placentomes towards more C and D placentomes. At no point in the control animals at day 90 did we see any C or D type placentomes. So we quantified this, and basically what we found is a significant decrease in type A placentomes and a significant increase in B, C, and D placentomes. Okay. So this was really interesting. I had no idea what this meant. All I knew was that we gave the animals testosterone, a lot of testosterone, and this is the consequence. The placentomes change morphology. Now, one thing that is a caveat here is even though the shift from type A to type D placentomes has been recorded in other sheep models of abnormal pregnancy, for example, undernutrition or overnutrition, if you will, inducing intrauterine growth restriction. We see this shift as well. However, there is no evidence whatsoever that a type D placentome functions any different than a type A placentome. So what I mentioned earlier about this fetal overgrowth is more a morphological observation than a functional observation. So this got us shifted thinking a little bit about, okay, we gave testosterone during pregnancy and we see an abnormal looking placenta. So what do we know about testosterone during pregnancy? So as most of you know, usually when we think about pregnancy, we think about estrogen levels go up and progesterone obviously is necessary. But what about testosterone? as it turns out that maternal levels of testosterone also increase during normal pregnancy in humans, but also, as you can see in this graph on the right, in sheep. And in fact, in cows, it's the same thing. So during pregnancy, it's not only estrogen, progesterone that goes up, but testosterone levels go up in blood as well. Women that, for example, have preeclampsia or polycystic ovarian syndrome or gestational diabetes, which are all associated with an abnormal pregnancy and placental dysfunction, are more likely to have increased level of testosterone. So, and finally, on top of that, is there have been some older studies that have been done that indicate that the placenta itself can secrete and synthesize testosterone. So not all the testosterone coming from either mom's adrenal gland gets aromatized by the placenta. The placenta itself can actually secrete testosterone. So it's one thing that it secretes testosterone, but we saw was a change in the placenta itself in response to testosterone. We started asking questions like, are there receptors for testosterone, like androgen receptors present? So androgen receptors have been reportedly present in the human and cow term placenta. There's a correlation between androgen receptor functional losses in terms of its expression with multiple miscarriages. Polymorphisms in androgen receptor are associated with preterm birth and abortions. And androgen receptors are increased in women, like I said before, with gestational diabetes. So the fact that the androgen receptor is present in the placenta indicates that the placenta is a target of androgens. Okay, so it not just only synthesizes testosterone, it's also a target for these androgens. So basically the big picture in our lab is to look at placenta function during pregnancy. Lots of things can negatively impact placenta function and lead to placental insufficiency. If you have placental insufficiency, we have less nutrients and oxygen going to the fetus, and we get things like intrauterine growth restriction. Intrauterine growth restriction happens in about 5 to 15% of pregnancies in the US and in Europe, and are associated with preterm birth, postnatal cardiac dysfunction, impaired neuronal development, and metabolic and cardiovascular disease. The three that I highlighted here, preeclampsia, gestational diabetes, polycystic ovarian syndrome, are all associated with abnormal testosterone levels of abnormal presence of androgen receptors in the placenta. So the overall goal of our lab right now is that we shift gears a little bit from looking at fetal gonads and microRNAs, or looking at exosomes and follicle development, and really focusing on determining the role of what really does testosterone and androgen receptor do in the placenta. We have a hypothesis that it potentially regulates angiogenesis, and the reason why I believe that is what I'll show in the next couple of slides. So sheep as an animal model, we've used sheep here at the Animal Reproduction and Biotechnology Lab for many years. Like I indicated, it's very cost effective as a large animal. It serves as a great model for cattle. It's used extensively as an in vivo model for pregnancy and placental insufficiency in humans, but again, also in ruminants. Placental and fetal physiology between humans and sheep match very well. I'll show you a picture of a placenta in humans and in ruminants. Even though morphologically these things don't look alike at all, in terms of how they work and how they function and how nutrient exchange occurs, it is very similar. And obviously, if you compare this to what is going on with mice, mice are one, a litter-bearing species, and two, sampling in mice is very difficult. However, in sheep, introduction of fetal catheters in a variety of places has been done a lot, and you can actually continuously sample blood during pregnancy and look at changes of blood flow through the placenta. So, like I indicated, for those of you that have not done or looked at placentas much at any one point, the human placenta is really a discoid placenta, and discoid because it's really a disc shape. So we have one disc unit where we have exchange of fetal and maternal material. Okay, this is indicated by all these chorionic villi, or these little tree-like structures that are highlighted right here. I just noticed a question from on YouTube. If we've looked at the histological sections of those placentomes between type A and type D. We have not. Other people have published on type A and type D placentomes. And that's why it does look like there's more of the cotyledon part instead of gross morphology-wise. Histologically, this is similar. You see more of the fetal tissue, more of the cotyledon tissue. So there is the assumption that there's like an overcompensation on the fetal side for this a little bit, where maybe there's initially impaired flow to the fetus, and the fetal side tries to compensate by increasing its surface area. But again, this is pure speculation. Nobody's ever placed anything across or measured anything going across a type A versus type D placentome to see functionally if there's a difference there. So going back to the human placenta, really there's different cell types here. So this here is the fetal side, these chorionic villi, which is all the fetal portion of the placenta is bathing in maternal blood. Key here obviously is that fetal and maternal blood needs to be separated. We cannot have mixing of fetal and maternal blood. And so in this case, we have cytotrophoblast cells that lie underneath a syncytiotrophoblast layer. The cytotrophoblast cells are proliferative cells and stem cells. Those are the cells that will continue to proliferate, turn into extravillous trophoblast cells that migrate and invade spinal arteries of the uterus, kind of opening them up, replacing the endothelial cells, so basically enhancing blood flow. The syncytiotrophoblast layer that covers these chorionic villi is a large syncytial tissue. It's a layer. It's a fused syncytium where we have nutrient exchange happening and hormone secretion. So this is where most of the hormones come from. Now, when you compare that to a placentome from a ruminant, it is quite different, okay? This would be a side view of one placentome. The really light-colored area here is the fetal portion with some fingers going down. So this is the cotyledon area. And the dark portion here going up, again, some finger-like extensions, that is the caruncle tissue. And in this case, we have, again, no mixing of mom and fetal blood, and the blood supply is separated by several layers. So in the cartoon picture here, basically what happens here is we have trophoblast cells that undergo incomplete cytokinesis, and they form binucleate cells. These binucleate cells, think of it as what the word says, it is a cell with two nuclei, and they actually can form giant cells. And according to some recent work by Greg Johnson in Texas, they postulate that these giant cells or these binucleate cells actually can start replacing the epithelial cells of the uterus. So this is kind of a form of invasion as well. So we do know that these binucleate cells definitely start fusing and forming this syncytium layer that also is important for nutrient transport and exchange. So a question again, how about the interdigitations at the fetal-maternal interface? This has not been looked at histologically in type A or type D placentomes. That's something definitely worthwhile doing. The problem is that interdigitation actually becomes quite, it's very tight and close, because a lot of times what we try to do when we collect placentomes to study the fetal versus the maternal portion, you can try to separate those. And basically what you're doing is you're peeling them apart. But this is very crude, and you always leave some maternal tissue behind in the cotyledon, and vice versa, cotyledonary tissue behind in the caruncle. So basically you're ripping these layers apart. But with the fusion of some of these cells and fusion of these binucleate cells with either the epithelial cells of the uterus to form this syncytium layer, so a fetal-maternal syncytium, or according to this new hypothesis that the epithelial layer is phagocytosed, those layers really start sticking together. So one of the things we knew that androgen receptors were reportedly present in the human placenta, in the term placenta. So one of the things that we did was we looked at first trimester placenta, because in order for androgen receptors to play a role throughout pregnancy, we needed to make sure that androgen receptors were present early on as well. So this is from a paper published last year, looking at androgen receptors in first trimester placenta. We also did some immunofluorescence. And basically, the androgen receptor appears to be present marginally inside the trophoblast cells, and this matches some other papers that have been published as well. They're present in the syncytial trophoblast layer, but do not really appear to be present in extravillous trophoblast cells. What we didn't know at this time was if androgen receptor is present in the sheep placenta. If we want to use the sheep as a model for human pregnancies and also for ruminant pregnancies and studying androgen receptor, this is something we absolutely had to know. Otherwise, there's no point of doing any of this stuff. So nobody had reported the presence of androgen receptors in the sheep placenta up until this point. The first question we asked was, is it present? And then, is it present during early stages of pregnancy? And the reason why we wanted to start looking at the early stages of pregnancy is because ruminant early development is so different from mammalian development. As most of you familiar with ruminant reproductive physiology probably recognize, is that early embryo development consists of forming a morula, a blastocyst that eventually hatches at about day 12, maybe a little later, and then it starts elongating. Okay, so when it starts elongating, it expands, and it basically forms this long tube-like embryo. Most of this is all trophectoderm. And this embryo, in fact, continues to elongate to a point where we have something that can be close to 20 centimeters in length, okay, containing an epiblast or an embryo and all this trophectoderm. So early embryonic development is characterized by an enormous amount of proliferation of the trophectoderm before it starts attaching to the maternal side, to the caruncles, and forming these placentome areas. So the first thing we wanted to know is, in this early embryo, as it starts elongating, is it already present? So we did some immunohistochemistry. So this is what a day 16 flushed embryo looks like. Most of this, again, is trophectoderm. And we can see clear nuclear staining of androgen receptor throughout the trophectoderm in these embryos. So we also looked at day 75. So this would be mid-gestation placentome. So on your right is immunohistochemistry. It's present throughout. So this would be the maternal side, where you have thick maternal stroma. And this is the fetal side, where it's a little bit looser stroma. And here's the interface between cotyledon and caruncle. And the cotyledon area, or the fetal area, is shown right here. We have a couple of binucleate cells right here, and they all stain positive for androgen receptor. Immunofluorescence also shows it nicely. What we show here in green is nuclear staining for androgen receptor, also present in maternal stroma. And what we used here for red is lectin. And lectin is a marker for fetal trophoblast cells. So what is highlighted here in red are all the fetal trophoblast cells, and we see green binucleate cells and fetal trophoblast cells. So that's all good. That means that the androgen receptor is present early on in pregnancy in the placenta. It's present in mid-gestation. And now we started asking questions like, okay, are they involved with some of these changes in the placentome? The first thing that a former graduate student, Ellie Claes, in the lab did, she wanted to just ask a very broad question. Are there any changes in DNA methylation in the placentomes? And so we compared control type A placentomes with type A placentomes from testosterone-treated animals, and we saw a significant decrease in global DNA methylation. Now, this is interesting, but it's also not very informative because we don't have any idea where the changes in DNA methylation occur within the genome. However, it does suggest that if we give androgens, lots of androgens, that we see hypomethylation, global hypomethylation in the placenta. When we repeated this in an ovine trophoblast cell line, we saw the same thing. This is a control amount of global DNA methylation in OTR cells. So OTR stands for ovine trophoblast cells. When we treat with testosterone, we see significant decrease, so hypomethylation. And then when we treat with testosterone and flutamide, an androgen receptor antagonist, so blocking the effects of testosterone, we alleviate this hypomethylation, DNA methylation effect, suggesting that this global DNA hypomethylation is due to androgenic effects. We looked at, Ellie wanted to look at lots of things involved with epigenetics. So H19 is an imprinted genes. We saw a significant increase both in the fetal and the maternal side of H19, this imprinted gene. DNA methylation itself was not altered in the cotyledon or caruncle, but protein levels were significantly increased for DNMT1, the DNA methyltransferase 1. She also started looking at something called KDMs. KDM stands for histone lysine demethylases. So histone lysine demethylases, an example is KDM4C here, showed a significant decrease on the fetal side. And the reason why she became interested in KDMs was the fact that a lot of these KDMs interact with sex steroid receptors. A lot of this came from studies in cancer cells. So these lysine demethylases basically demethylates a specific lysine residues on histone 3. So this is a large family. I'm only showing half of the family really here. But KDM1A, 1B, KDM1A specifically interacts with androgen receptors and estrogen receptors. So they form protein dimers. KDM3A, 3C as well, KDM4C. So, and all of them have specific targets on these lysine residues on these histone tails. And the idea here is that basically this lysine demethylase, depending on what they demethylase, either opens or closes chromatin and allows access to promoter sites or not. So we started pursuing KDM1A a bit more. The reason for that was looking at the literature, there was some evidence that in fact KDM1 or KDM1A is necessary for global DNA methylation. The fact that we saw a decrease in global DNA methylation suggested to us that KDM1A somehow may be impacted here. On top of that, is a paper had been published in Nature Communications where they knocked out KDM1A in trophoblasts, and they found that there was problems with trophoblast differentiation. So in this case, we were interested in KDM1A because one, it is probably important for placental development, and two, it interacts with androgen receptors. So the way it interacts with androgen receptors is shown in this cartoon picture here, where upon testosterone binding to its receptor, we get dislocation of heat shock proteins, dimerization of ligand-bound androgen receptor, movement to the nucleus, and now we get interaction with KDM1A. KDM1A will then demethylase certain lysines, and we get transcriptional activation. Depending on the context here, whatever gets demethylated, this could also lead to gene repression. It really kind of depends whether the lysine is lysine 9 or lysine 4. So the first thing we needed to find out was if KDM1A, in fact, is present in human in sheep placenta, because up at this point, we had no idea. So again, we have term placenta here, first trimester placenta here, and KDM1A protein present within the placenta. These are two human trophoblast cell lines. They also express KDM1A. We also looked at the sheep placenta. Similar again here. This is the fetal layer. This is a binucleate cell. You can see clear staining of KDM1A in the fetal portion of the placenta as well. It's also shown in this confocal imaging. Lectin here stains again the fetal side. The maternal side is right in the middle. And you can see KDM1A is present in the fetal trophoblast cells as well as some of the maternal stromal tissue. So next question is, okay, KDM1A and androgen receptor complex together in cancer cells. Do they do so in placenta? So co-immunoprecipitation assays were done showing that indeed we can use a KDM1A antibody to pull out androgen receptor, and we can also use an androgen receptor antibody to pull out KDM1A. So within sheep trophoblast cells, these two proteins complex together. We also did the same thing in human trophoblast cells, and we find the same thing. Again, KDM1A and androgen receptors are physically interacting in human trophoblast cells. So then we started thinking, okay, how can we study KDM1A function, and does it do anything if we knock it out? So we ended up using CRISPR-Cas9 technology to generate a human trophoblast cell line without any KDM1A. So this is scramble control. This is the cell line where we mutated KDM1A, and KDM1A protein is absent, as shown right here. When we look at androgen receptor levels in these KDM1A knockout cell lines, we see pretty much a complete abolishment of mRNA levels and significant reduction of androgen receptor protein. This suggests that KDM1A, in fact, may be involved with regulating androgen receptor expression as well. The reason why we started focusing on angiogenesis, because angiogenesis is one of the hallmark features of placentation. You need angiogenesis to happen. Without angiogenesis, we cannot share nutrients or exchange nutrients whatsoever. So VEGF is one that's really important in human placentation. It's important for cell proliferation, migration, development of blood vessels, and in the sheep placenta as well. So this is a nice review figure of what happens on the fetal side with angiogenesis versus the maternal side. So on the maternal side, during placentation, we see more, the branches are getting larger and wider. In the fetal side, we see more branching itself. So we looked at androgens, if they could actually upregulate VEGF expression as a marker for angiogenesis, and human trophoblast cells exposed to dihydrotestosterone significantly upregulate VEGF-A expression. And when we looked at those sheep placentomes that had undergone prenatal androgenization, at gestational day 90, when we collect these placentomes, we saw a significant increase in VEGF-A protein in both the fetal side as well as the maternal side. So this suggests that testosterone treatment is involved with regulating VEGF in placentomes and in human trophoblast cells. So can we do actually knockdown of KDM1A in the placenta in vivo? And this involves placenta-specific gene targeting. And this, in fact, is something that we do right now at CSU at the Animal Reproduction and Biotechnology Lab. So the idea here is that we take a day 9 sheep hatched blastocyst, we infect it with a virus containing shRNA constructs or CRISPR-Cas9 constructs, and we incubate that for 4 to 5 hours. That virus will only infect the trophectoderm. None of this goes to the inner cell mass. So basically the red here indicates trophectoderm that has been infected with a target construct. And in this case, we start knocking down or knocking out, if you use CRISPR, the gene of interest. We then surgically do embryo transfer surgeries to recipient use and then look at the effect on placental development. This originally was developed in mice. This is a great picture that shows that if you infect a blastocyst, that only the trophectoderm is impacted. So in this case, they used the GFP construct. The embryo is not green. The placenta is green. And subsequently, we've used it in sheep, to knock down Lin28. Dr. Anthony used it to knock down a variety of genes. And so we, 2 years ago, started doing a pilot study to use the CRISPR-Cas9 construct to knock down KDM1A in the sheep placenta. So again, day 9 embryos were infected. This is to show that the KDM1A was able to completely knock down the KDM1A protein. This is a control elongated day 16 embryo, and this is one of the embryos that we were able to collect following knockout of KDM1A in trophectoderm. So KDM1A in that trophectoderm is significantly lower. Androgen receptor is significantly downregulated in these trophectoderm cells. So basically what we're doing right now, actually today is one of our surgery days. So this morning, everybody was in the barn at 6 o'clock in the morning. We flushed a whole bunch of embryos. And as soon as I'm done with this presentation and meeting with you guys, I'm going to go back to the surgery room and we're going to surgically transfer recipient embryos that have been infected with a target construct. Today, we're trying to knock down androgen receptor. So a graduate student, veterinarian, Dr. Agatha Parsons, is involved with that project. A master's student, Taylor Horde, is involved with knocking down KDM1A in vivo. So it sounds like I do all this stuff. Juliano knows this way better than I do. I am barely ever in the lab. The students actually get to do a lot of the fun stuff. Although I do get to flip some sheep early in the morning. So really, the androgen receptor KDM1A project involved a lot of people that have moved on. A PhD student, Ellie Clays, Jennifer Halloran, Aaron McWhorter, Rachel, Jennifer Russ, Ali, who recently graduated, a lot of current graduate students on the project, and lots of collaborators. And it's really nice when I put this slide together to indicate that I can now say Doctor. I'm not ready yet to say Professor Dr. Juliano da Silveira, but Professor Dr. Juliano da Silveira, who's also been involved in the early stages of these projects. And we've been fortunate to get funding for this. So right now we're 2 years into a 5-year USDA grant that funds this project. So with that, I really like to thank you for attending this seminar. I hope it was something interesting and educational. And if you have any questions, let me know.
Thank you, Jerry, for the talk. It was an amazing talk. It's really nice to see all the data that you put it together in the last years. And so it's nice to see. So I will send a question here in the chat, but I can read to you before. And please, everybody that's watching, if you have a question, just send, and we are going to show up for Jerry. So I thought it was interesting the way that you showed it up, that when you knock down KDM1A, so you had no protein levels of AR. So did you also check the gene expression? So that means that KDM regulates androgen receptor gene expression, or could we guess some change in localization, for example, non-nuclear to cytoplasmic?
Yeah, when we started noticing that, we did try to look and see if we can actually use chromatin immunoprecipitation and see if we can use a KDM1A antibody to pull down androgen receptor promoter, the androgen receptor promoter, and we could. So KDM1A protein actually sits on the androgen receptor promoter region. So it is involved with androgen receptor expression regulation.
So, and just to another question. So have you guys thought about doing chip-seq of these, which genes are regulating, you know, when you have KDM and AR all complexed together?
Yeah. So this at the end of the year, we'll be finished with 2 years of the grant, which was mostly focusing on developing those target constructs. So in the subsequent years, that's actually one of the experiments that's proposed in the AIMS-2 is actually, exactly what you said, is to do chip-seq and find out exactly the genes that are regulated by KDM1A in the trophectoderm. And actually the plan is to separate XX from XY cells. So some of the data that I didn't show is that in fact there is a difference in gene expression of KDM1A between XX versus XY. Trophectoderm cells. So yeah, we still have a long way ahead. These 2 years went by way too fast. So Jerry, I would like to thank you one more time for your talk. Everybody's saying it's thank you through the chat. You can check. So we had pretty much around 40 people watching the seminar. Awesome.
And our audience, it fluctuated a little bit. Right now we have 31 people online yet.
Yeah.
And so it was a pleasure to have you here. I don't want to hold you from the fun part of the research to go to the lab.
Yeah.
And I really appreciate. So if you want to give a couple last words before we say goodbye. So
thank you so much, Juliano. I really appreciate it. It's always, I really enjoy presenting some of the work that we're doing. And there's a lot. We're kind of in the middle of all these experiments, and I can't wait next year at some point to come out and really present some updated, more new information of some of these projects. Throughout the years, as you know, we've always enjoyed all our collaborations, having students come out here to work on projects. Obviously, the whole COVID thing is kind of problematic right now, but I really hope that in the future we can continue doing some of this collaborative stuff. It'll be great.
Yeah.
Definitely really miss you flipping sheep.
So yeah, I forgot to mention, but Dr. Bouma is one of the speakers for the next SBTE. So he was supposed to come this year, but because of all the changes, we decided to postpone his trip for next year. So next year he'll be at speaking at SBTE. So
Yeah.
hopefully in a nice place. Huh?
Yeah, I can't wait. I really look forward to it.
All right. Thank you, boss. And
Yeah.
I'll see you hopefully soon.
Yeah, try to get some sleep.
All right. Thank you, everybody, for participating. And let's try to keep this going. So we'll organize other talks and hopefully invite all of you. Bye-bye.
Yes. Thank you, everyone. Really appreciate it.